Well, hello. In today's lab, we are going to look at osmoregulation in invertebrates. And the point of this lab is to see how these different types of marine invertebrates cope with different salinities of seawater. So what we're going to do here is we've got an array of seawaters of different concentrations, going from 0%, which is basically deionized water. There's no salt in it, no ions. 25%, 50%, 75%, 100% seawater, so about salinity of 35 parts per thousand, normal seawater, and then a more concentrated seawater solution that's at about 150% seawater. Okay, so the way we make these solutions, of course, deionized water we get downstairs from my distillery. 100% seawater I have shipped in from the Bering Sea every Thursday night. And the other concentrations we have to make by adding salt to deionized water in the correct proportions. So that's what I'll do now. Just to give you an example, so we've got some fresh water, we're gonna put some salt in. Now normally, again, in a lab, we would be wearing protective equipment and things, but in this case, we're just using regular old table salt. So that's not, oh God, oh, it burns. My eyes, cut. Remember that animals can generally be classified as osmoconformers or osmoregulators. Osmoconformers, like the figure on the left, shows that as the external environment changes, the animal's internal environment changes with it to match whatever the concentration of the external environment is. So they're not really fighting the environment, they're just letting their body tissues equilibrate with whatever's external to their bodies. A regulator, on the other hand, tries to keep its internal conditions constant regardless of what's happening in the external environment. So in the figure on the right, you see the zone of stability, where it's maintaining homeostasis. Regardless of what's happening in the external environment, the internal conditions are remaining fairly constant. So in this experiment, we're going to look at how different invertebrates react to changes in salinity. Do they show the typical conformer line like we see on the left, where their body tissues match the external environment by taking in or losing water? Or are we going to see the regulator curve that we see on the right, where regardless of the external conditions, the animal is going to maintain consistency internally? It's not going to lose or gain any water. So the animals that we're going to use in this experiment are two different kinds of marine invertebrates. And first off, we have clamworms, who are a polychaete, soft-bodied worm, a benthic species. Oh, I think this one's dead. So let's get another one in there. We want to make sure they're all alive for the experiment. And the other type of animal we're going to use is a crustacean. We're going to use a little crab. Right now, so both of these are marine organisms. They are used to tolerating seawater and a wide range of possible salinities. So we're going to see how they react in these different seawater treatments. So the first thing we need to do, because we're trying to measure mass loss or mass gain in these animals, is weigh them. So I have my accurate scientific balance here, and we weigh them to get their first initial weight. And this looks like it's 1.37921 grams. Okay, so we take note of that, weigh each of the animals, weigh the crab. Now, I'm sure you're expecting me to do some kind of joke about being pinched by the crab here, but I'm not one for cheap laughs, cheap theatrics. This is serious. So we weigh that one, and now we have our initial weight for these. So now we're ready to put our animals into solutions. Okay, so now that we have all our animals weighed, we know their initial masses, we've got all our water samples here with our known concentrations. All we need to do is put the animals in their respective beakers. Okay, so now a worm is going to go into each one of these seawater beakers. We've got the initial mass, we've noted the time, and now we're going to let nature do its thing with each of these worms. Over the next hour, we're going to measure about every 15 minutes, take them out, reweigh them, and see if there's any change in mass. Right, for the crab, usually we don't have a whole lot of crabs, so we just use one thing of water and gradually, every 15 minutes, put the crab into a different solution, different concentration of water, and see what happens to the crab. Okay, so crab goes in there, And osmosis, do your thing. We're going to wait now for the next hour, measuring every 15 minutes, taking another weight, and see what happens. Okay, so we'll see you in an hour. Okay, well that's been an hour, so now we can see how these little critters are doing. And so we're going to get our scale once again, and now for each of these, of course I was weighing them every 15 minutes until now, so now we're getting just our final 60-minute weight. And we can weigh them and take note of the weight of each one of these. And from that we're going to calculate percentage mass gain or potentially mass loss, or is there no change in mass? And depending on how these animals react, do they gain water, do they lose water, is there no change? That's going to tell us something about how they deal with different concentrations of seawater. And we can then determine, are these animals regulators who are keeping their internal conditions as constant as possible, or are they conformers who just let their internal body conditions fluctuate with whatever the external environment is doing? So how much water they take in or lose will indicate something about, are these animals conformers or regulators, and how do they deal with these different saltwater solutions. So now you have your data. The next step is for you to run your analysis and complete the write-up. See you next week.